Neonatal period, structural organization. The development of gyri and sulci continue with formation of tertiary convolutional patterns. The prefrontal cortex is cytoarchitectonically immature, and the main reason for this immaturity is that granular layer 4, a main recipient of thalamocortical input, remains well developed in all frontal areas, that is, in both prospective granular and prospective dysgranular areas. In layer 1, the formal fetal marginal zone, there is concentration of neurons and well-developed Cajal-Retzius cells are still present. The newborn cortex has immature laminar organization with prominent layer 4, dense layer 2, absence of magnopyramidality of layer 3C, and wide transition between layer 6 and the white matter. Cytoarchitectonic remnants of the subplate zone, figure 2.4. The precise delineation of dysgranular areas, Broca's area 44, posterior orbital cortex, from prefrontal granular areas is not possible on the basis of cytoarchitectonic criteria. Neurogenetic events. The neonatal period is dominated by dendritic differentiation, figure 2.5. Growth and reorganization of callosal fibers and growth of short cortical fibers. Dendritic development is evident in all cases or classes of neurons. At birth, layer 5 pyramidal neurons have larger and more complex dendritic trees than those of layer 3C. Pyramidal neurons of layers 5 and 3 display the first phase of spinogenesis and intense growth of basal dendrites. Interneurons of layer 4 develop characteristic phenotypes and can be distinguished. The supragranular layers contain double bouquet neurons, basket neurons, and multipolar and bituft non-pyramidal neurons. It is interesting that subplate neurons show continuous dendritic growth. The growth of short cortical pathways was not directly demonstrated in human prefrontal cortex, but in the human visual cortex, DII tracing demonstrated growth of short cortical pathways during corresponding phases of development. And then that DII is capital D, lowercase I, capital I, and I found a reference for 1990 neuronal tracing with DII, decalcification, cryosectioning, and photoconversion for light and electron microscopic analysis, with authors von Barthold and Rubel and Cunningham. And this is the molecule 1, 1 prime dioctadecyl 3, 3 prime, 3 prime tetramethyl lindocarbocyanine perchlorate, or DII, is a fluorescent dye which diffuses within cell membranes. The properties of DII diffusion and fluorescence are maintained in aldehyde fixed tissue, thereby allowing selective neuronal tracing postmortem. We describe 3 modifications of this tracing method. This is all the abstract of that paper. First, while DIL typo diffuses along neuronal membranes, the tissue can be decalcified in EDTA at 37 degrees Celsius. Tracing in decalcified tissue extends the possible application of DII technique to the investigation of neuronal tissue enclosed in bony structures. Never mind. Second, we describe a protocol that allows sectioning of DII injected tissue on a cryostat with minimal subsequent spread of DII in dried sections. Third, we demonstrate the DII label fluorescent neurons in cryosections as well as vibrotome sections can be photooxidated and converted to a stable diaminobenzidine reaction product. The photoconverted DII label is electron dense and allows analysis of labeled cell bodies and processes at the electron microscopic level. DII does not stay confined to the surface of the cell membrane and fixed tissue, but reaches internal organelles, presumably via membranes of the endoplasmic reticulum, and concentrates in microsomal structures adjacent to mitochondria. Photoconversion of DII label is compatible with gold immunohistochemistry. Long-term incubation and subsequent photoconversion of postmortem DII-labeled neurons provides remarkable tissue preservation at the ultrastructural level. Okay, back to the book. So I'm going to read that paragraph again. It is interesting that subplate neurons show continuous dendritic growth. The growth of short cortical pathways was not directly demonstrated in the human prefrontal cortex, but in the human visual cortex, DII tracing demonstrated growth of short cortical pathways during corresponding phases of development. Synaptogenesis is quantitatively significant neurogenetic event in the neonatal cortex, and there is rapid increase in the formation of synapses. So these authors are from Croatia, I believe. Croatia. So there aren't always articles like the and a in the writing. Functional organization. During the neonatal period, the lamellocortical circuitry is established, and there is substrate for interaction with other brain areas or other brain regions and sensory environment. This process is very important for shaping of fine connectivity within the cortical columns. During the neonatal period, the anatomical substrate of electrical activity is rapidly changing because of exponential increase in the number of synapses, deep synaptic activation, retraction of exuberant callosal axons, and ingrowth of cortical fibers. As a consequence, the cortical dipole changes and the cortical surface response becomes predominantly positive. The EEG shows more continuous activity. Reference Dreyfus-Brzak, 1979. Slow activity transients may be still present in the full-term newborn. This phenomenon, together with the prolonged existence of transient circuitry and immaturity of cortical connectivity, points to the overall functional immaturity of the cortex. Supragranular local circuitry neurons have non-myelinated axons and immature dendritic arborizations. This immaturity of neuronal circuitry correlates well with immaturity of the psychological basis for recruiting and sustaining attention to a stimulus that is guided primarily by its physical features.